There it is.
All right. Welcome, everybody, to another StaPH-B call. This one was a long time in the planning with both Robert, Tim, myself, and Sean. We've been wanting to hear this talk for a good time now. This is really a follow-up talk to Staphopia, as Robert will present. And for those of you who don't know Robert Petit is a senior data analyst at Emory University and their Division of Infectious Diseases. And yeah, so just taking a break, hopefully, from some of the SARS-CoV-2 madness to get back to some of the bacterial genome analysis and looking at this really comprehensive platform for general bacterial genomic analysis that has pretty much all of the QC, taxonomic, and other common public health bioinformatics applications built into a single platform, and also utilizing a lot of the same premises that we try to adopt here at StaPH-B in terms of containerization, workflow management, and everything in between. So with that, Robert, if you want to take over, I'll go ahead and mute my mic.
All right. Thank you, Kevin. Thank you, everybody, for being here on this Friday afternoon. I'll try to get through this quickly so we can get to the discussion. So yeah, I've titled this talk is Bacterial Genome Analysis using Bactopia. There we go. Briefly, I'm just going to introduce Bactopia, present a use case, and then give some behind the scenes of what's happening in the background that you don't really get to see in like the papers and whatnot. So what is Bactopia? I describe Bactopia as an extensive workflow for processing of Illumina sequencing of bacterial genomes. The goal of Bactopia is to process your data with a broad set of tools so that you can get to the fun part of analysis quicker. Because I think we've all kind of been there where you're slogging through the pipeline. You're just like, I just want the output so I can start analyzing the data, and not deal with the getting everything running and all that. So my philosophy when I developed Bactopia was, one, to be Conda first. Make sure all tools, everything that is used by Bactopia is available from an official channel, such as BioConda, CondaForge, or like defaults or R or whatever. And if it's not available, can I create a recipe? I chose official channels. Yes, there are some tools that are on personal channels, but those, if it's on an official channel, it's more likely to be maintained. If there's a problem that arises, there's going to be more people who can be like, oh, I can fix that real quick. Whereas if it's on a personal channel, I might have to track you down, or I may have to make a copy and then create one myself. I wanted Bactopia to be flexible and portable, so it should fit the user's needs. All the parameters for every tool that's used, you can adjust. There are over 100 adjustable parameters. I don't suggest you go adjusting all 100 parameters just because. I mean, if you want to test Bactopia, figure out the extreme cases or the edge cases, thank you, but it's just, there are use cases where I need to adjust some parameters for Proca or stuff like that, but most of the defaults are put by the developers and are good. So it should be easy to install. You can install Bactopia through Conda, just Conda install Bactopia. It's available on BioConda. It has a Docker container and Singularity container. And it should be easy to switch between environments. So whether you're running on your local machine, a cluster, or the cloud. And that happens thanks to Nextflow. And documentation. Too much is better than too little. And when in doubt, document it. And I think I've done a pretty thorough job of documenting Bactopia. If you see something missing, please let me know and I will put it on there. There are kind of three sides to Bactopia. There's Bactopia datasets, which is a framework for including public and user-created datasets. It's completely optional, but I highly recommend it because it will very much increase the value of the analysis. There's Bactopia analysis pipeline. That's the main per isolate workflow. And Bactopia tools, which are a set of independent workflows for comparative analyses. So to put it all in one little diagram, let's see, do I have, oh, go back, let me get the pointer. basically you take your inputs, that could be either from your local machine or from ENA or SRA. Bactopia will download ENA and SRA accessions for you and process them. You can include the Bactopia data sets, and at runtime, the analysis pipeline will determine what data sets you have available and then make the necessary, process them, include the necessary processes for those depending on the data sets available. Otherwise, it'll just run standard, no data set required analyses. And then you get your analysis results in a predictable data structure so that each sample will have the same output structure. And that way we can very easily see, find all the files that we need to find. There's no like random files here, random files there, and try to figure out where everything is. And then using this standard directory structure, Bactopia tools will basically use these structures to find the input files that it needs. For example, if you wanted to create a pangenome with Rory, the Bactopia tool for Rory will find all the GFF files for each of the samples and then create your pangenome. And then you get analysis reports. This is a overview of the analysis pipeline. To give you an idea, these in these kind of grayish squares, all these are always enabled. So these analysis steps, no matter whether you give data sets or not, are always enabled, because you don't necessarily need data sets to create an assembly. Whereas these orange circles or ovals, these require input data sets. So if you want to call variants, you need a reference genome, and the Bactopia data sets can help you select reference genomes. Or if you want to call sequence types, you need a pub-MLST schema. And these are just data sets that you can include. The blue diamonds are optional data sets that the user can provide, and the blue circles are data sets that can be downloaded automatically using the Bactopia datasets tool. So yeah, I won't belabor what's happening here. I can circle back at the end, but this is kind of like what I would consider a standard bacterial genome analysis pipeline, everything that you might want in it. So to present a use case, we decided, hey, you know, we're working on some vaginal microbiome studies. Let's take a look at all the Lactobacillus genus, and let's just run them through Bactopia. So yeah, we'll run all available Lactobacillus genomes through Bactopia. This will demonstrate how to build Lactobacillus data sets. This will demonstrate the use of querying ENA for available Lactobacillus genomes. This will show how we can download directly from ENA or SRA. Just assume those are interchangeable. If I say ENA, I'm also meaning SRA. And how we can download those samples directly and then apply each of the tools for sequence quality summary, a 16S phylogeny, and taxon classifications in a core genome on just a subset of the samples. So yeah, we downloaded about 1,400 samples. At the time, there were only 1,400 Illumina samples for Lactobacillus. It took about 2.5 days to process on a kind of like hodgepodge cluster that Tim and I put together using old servers around the lab. I think it's probably 96 cores total. And so yeah, most of the data, we classify the data as gold, silver, bronze based on coverage, quality scores, and read length and content counts. One thing that Bactopia does that Staphopia also did is we downsample to a set coverage. The user can set the coverage or disable this completely. But we found that after you get diminishing returns after you have so much coverage, but you really start to increase the runtimes analysis when you have high coverages, because it takes a whole lot more time to QC 1,000x worth of reads versus 100x, or it takes a whole lot longer to assemble 1,000x of reads versus 100x. So by default, it will down select to, and this is randomly, it basically just randomly subsamples reads from your data. It'll downsample to 100x. And since this is all Lactobacillus genus, the genome sizes in Lactobacillus range from, I think it was 1.2 to almost 4 megabases. So we couldn't, if we were doing like Staph aureus, we could just say set the genome size to 2.8 and use that to downsample all our data. So when we're estimating coverage, we need to know a genome size. And so since Lactobacillus, we didn't know the genome per se, we could estimate it using MASH. And so we did that to get this coverage, but we found that the MASH estimate is most commonly is much larger, not much larger, slightly larger than the assembled genome size in all our cases. There were a few where the assembled size was way larger than the estimated genome size, but those tended to have other issues, not necessarily the sequencing. So what did we find out when we ran Lactobacillus? So one thing we decided to do was we wanted to just create a 16S RNA gene phylogeny. We used PhyloFlash. I chose PhyloFlash because yes, Proca will pull out 16S genes, but it won't always find the 16S genes. So if you have a bad assembly, you may not get the 16S gene. What PhyloFlash will do is it will try to reassemble the 16S gene. And so it'll basically align to a 16S, a database of 16S genes, and then use those reads that map to create a spades assembly. And then there were only a handful, so there were only a handful of samples that this PhyloFlash was not able to reconstruct a 16S gene. But that's fine, it was not that big a deal. Then we used the GTDB toolkit to basically classify each sample. GTB is the Genome Taxonomy Database. And we found that there were 58 samples labeled as Lactobacillus in SRA that, based on GTB, are not Lactobacillus. 34 of these were Strep pneumoniae. 2 of these were yeast. And I don't know if this was just errors in data submission, but it kind of like opens up this, like we kind of saw this when we ran Staphopia on 44,000 staph genomes. There were a few that were kind of like, at that point, they were Staph argenteus, which was a recent taxonomic change. Like recently, when the data was submitted, it was Staph aureus, but after a few years later, it got reclassified as argenteus, but it wasn't reflected in the database. But I mean, that doesn't quite work when, you got 34 samples that are Strep pneumoniae. So there's issues with data submission that you just have to be aware of. We also found that 33% of the GTD classifications are in conflict with the NCBI taxon classification. And some of those they make sense. Again, some of those it's a recent change. So scientists got together and said, no, these should be called this, and they changed it. And the 16S database may have seen that change, whereas NCBI may have not seen that change. So some of those make sense, and it just kind of highlights that we still haven't quite figured out this whole name and stuff. There were 5 major groups of Lactobacillus that have been sequenced. 45% of all available genomes were from these 5 species. And what we were kind of interested in was Lactobacillus crispatus. And we used FastANI, again, another Bactopia tool that we've created for FastANI to basically, we ran all the samples with FastANI against a Lactobacillus crispatus genome, and we used that to subset all samples that had greater than 95% ANI to that Lactobacillus crispatus genome. And it's really, you kind of see them all right here. And then we said, oh, great, we got these Lactobacillus crispatus. Let's use the Rory tool to create a pangenome. So crispatus is commonly isolated from human vagina and the guts and feces of poultry. And our pangenome kind of recapitulates that. These samples, or this clade here, tends to be from human. And these tend to just kind of be poultry, and there are some human cases in there. And then we also were able to recapitulate known tetracycline resistance in these poultry samples. So only the poultry samples had tetracycline resistance, except this one human sample, which is interesting. This is from the gut of a human, so not sure, maybe they ate poultry, who knows. So yeah, for just to kind of highlight the use case with this, we demonstrated how Bactopia can build data sets. There is a command, Bactopia datasets, to do this for you. Query ENA for publicly available genomes. There's a command, Bactopia search, that will take your query and query ENA's data warehouse and return the results for you. Process publicly available genomes just using the standard Bactopia command, and then conduct comparative analyses with Bactopia tools. So we have a summary report. The summary tool is pretty convenient because it creates a list of samples that you should probably exclude. So you can, when you run the summary tool, you can set some thresholds that you say, I want at least 20x coverage, or I don't want samples that have 1,000 contigs for downstream analysis, because those are probably samples that may have an issue that I should probably check out, or I don't need those. There's also the 16S phylogeny, which we use PhyloFlash and GTDB, and then the core genome on a subset of samples. So we showed how you can use FastANI to find the genomes that you want, or find the samples that are closely related to the reference genome that you have, and then create a pangenome. Because it wouldn't have made sense to create a pangenome on all of Lactobacillus. We probably would have just gotten a handful of genes, whereas we were interested in crispatus, and we showed how you can programmatically find crispatus genomes, because we also showed that you might not necessarily be able to trust the taxon classification that's available publicly. And that's to no fault on NCBI. There's a lot of wheels turning when it comes to submitting data and all that. Yes, this is available as a preprint right now on bioRxiv. So basically what I've just discussed, we kind of highlight in this, and we also give the commands that we use to reproduce this. So yeah, what's happening behind the scenes in Bactopia? Back to this overview. Right here, it's like, oh, there's not much happening. Now, when we add kind of like what's actually happening, Bactopia allows singled in and paired in FASTQs. You can download from SRA, ENA. You can also provide single or multiple samples. Sometimes you just want to process a single sample and you don't want to create a config file for that. You just want to say R1, R2 process. But also, when there's thousands of genomes, me personally, I don't want to figure out where your data is. I want you to tell me where it's at. I did create a tool for that, Bactopia Prepare, which you give a directory and run Bactopia Prepare, and it will create this file of file names for you, based with hard-coded paths. So that way it's easy to find stuff and all that, and there's nobody's trying to figure out where the data is. Bactopia data sets includes more than 10 publicly available data sets that can be downloaded and automatically set up for you. It'll also, if you give a species, download REST-seq genomes and create cluster proteins for annotations. So basically, you'll create a kind of like a set of genes that Proca can use for annotation first before Proca uses its default databases. This is kind of useful because then all your genomes will be annotated with the same set of genes, which are reflective of the species that you're currently looking at. It'll also create a sketch of those REST-seq genomes that it downloaded for automatic reference selection if you want to just call variants against the closest genome. And the user can provide their own reference genomes and their own genes and proteins that they may want to have blasted or call variants against, or just have reads mapped against. So for example, if you're interested in a specific gene and you're curious, does it actually have coverage? And if it does, is it uniform coverage, or is it something where I need this particular region of the gene, and when I do my coverage plot, oh, that gene's missing, stuff like that. The analysis pipeline, it's Nextflow powered. It includes over 20 different analysis steps and more than 60 tools. And the tools, the way I've set up Conda is I build from environmental files, so little YAML config files. I don't actually do the Conda install each time. There's a set of, I've already pre-built the environment. Here's the programs that need to be installed. This is convenient because it tells you which program version you know specifically. If you use Bactopia version whatever, you can go on the GitHub repo and see at that version it was using these program versions. But I also create a Bactopia versions tool, which will basically spit out what program versions are used in Bactopia at that point in time. And the analysis results, like I mentioned, it has a standard structure, so every sample gets, all the outputs get put in the same sort of data structure. And one thing is I've taken all the outputs and given a brief description of the output file. So yeah, so like Proca again, like it produces a lot of files, and I've basically taken like a one sentence, what this file, this extension contains this. I think that'll be helpful for users who may be getting started, saying, what the heck are all these extensions and what do these files mean? Now you can go to the Bactopia documentation, and I've kind of aggregated it all into a central place. Bactopia tools is also Nextflow powered, but these are all comparative analyses. So these are the type of analysis that you want to do on the complete set, not on the per isolate. It auto finds the inputs because of the standard structure, but you can also include or exclude specific samples. So like for example, when we ran FastANI to find the crispatus, I can create a list from that and tell Rory to only include these samples. So it can go to your directory and say, I want X, Y, and Z samples, and just get rid of A, B, and C. And for most of the tools, you can include completed genomes. So if you wanted to also include all crispatus completed genomes in your Rory pangenome analysis, that tool will go and download those genomes from the NCBI assembly database. And so yeah, the analysis reports can be used to aggregate all the results of these into one simple, one like table of results that you can take a look at. And it's also useful to find those samples that may have some data QC issues. Those samples that are like, that's weird, we should probably check that out. For like, why did it estimate a genome size of 12 megabases when I expect 4 megabases? And so for today, I will just keep it brief and talk about some things about Nextflow and how I selected the tools I use. First, I'll do tools. So does Torsten Seaman, is it part of the Torstiverse? Does he have a tool for analysis X? If yes, include it. For example, shovel, proca, and snippy. So yes, that's kind of a joke, but Torsten writes really good tools, like plain and simple. But for example, I've worked with like I've edited proca and shovel, so I have a branch of shovel, and it includes single end reads. So the main branch of shovel only includes paired end reads. But the one thing I like about Torsten's tools, if I've worked on proca, his code's going to be very similar for shovel, and so that's nice that when with his tools they're kind of similar, but he also writes really good tools. Jokes aside, I mean, is it a well-known tool, and is it a well-used tool? If yes, it should probably be included because the user's probably going to want to run it, and if you can include it in this one run, go for it. Is the tool still maintained? I put maintained in quotes here just because it's very loosely. What I mean by maintained is if the tool, I'm not saying is it still under active development. It's more is if a tool runs and does what it's supposed to, great. But if the tool, for some reason, there's a new update which causes this tool to no longer work, we saw that, especially since I'm in the Conda boat. Like I can't jump off the Conda boat now because I put all my eggs in the Conda basket. But if the tool is still maintained, if that break happens, if I submit a GitHub pull request or submit the issue, is it maintained enough that the author of the tool might see it and be like, oh, I can fix that real quick, and then done. It's like it's very loosely like I'm not looking for this is getting five commits per day and all that yada yada yada. Just is it just somewhat passively maintained? Is the tool Conda installable? Because like I said, I put all my eggs in the Conda basket. Like I can't escape it now. If not, can I create a recipe for it? And I have done that for a few of the tools. And kind of if there isn't a recipe, I kind of go down this. I mean, is the tool easy to run? Is it worth creating a recipe for? If it's one of those where like you run it and there's like no kind of like interaction, then maybe it's not the best, because this is also a tool someone else is going to use, and I'm trying to make it to where it's easy for everybody. And so also, is this tool a comparative analyses? If it is, it probably should be a Bactopia tool and not in the main analysis pipeline. And so Bactopia, it's powered by Nextflow. So why did I choose Nextflow over SnakeMake over XYZ? So this happened, oh gosh, 2015, 16 maybe, when I chose to, I sat down and I was like, am I going to use SnakeMake or Nextflow for Staphopia? And at that point, it just came down to if I have problems using one, because personally I was like, I know Python, I should use SnakeMake and go. But then I was also like, if something breaks, do I have to fix it, or can I just say go here? And go here being, it ended up, I kind of at that point is kind of like Nextflow. It had much better documentation, and this is not reflective on SnakeMake at this point in time. This is reflective on SnakeMake a few years ago. Nextflow has pretty good technical support. So if you want to, you can probably reach out on Gitter and contact one of the main developers of Nextflow, and they can help you directly. But there's on the Gitter chat, there's usually a number of people who are willing to help. You can also submit issues on GitHub, and they're pretty responding on Twitter as well. And Nextflow it does very well quickly switching between environments. So you create a profile for your AWS or your Google Cloud or your local cluster or your local machine, and just a quick parameter change, you're switching from your local machine to a cluster. It has resumable workflows, and at the point in time, it looked to be like it was well funded and it was going to be around for a while. And that's, to me, useful because I think when we kind of make these type of tools, we don't necessarily want to be the ones that have to maintain like the workflow managers or like specific tools. Like, I'm more gluing everything together. I don't necessarily want to be the main maintainer of each of these tools. So basic Nextflow structure kind of looks like this. You have inputs, and then the inputs go into what's called a channel, and then the processes are kind of where the analyses happen. So that's kind of like your shell commands and all that. And then you get some outputs. So this is just to give you a brief introduction to kind of show some of the gotchas that I've run into with Nextflow. So there's two types of channels with Nextflow. There's a queue channel, which is a first in, first out queue that connects two processes, and it can only be used once. So another thing, this reflects, there's a newer version of Nextflow that will know, basically you can use channels more than once in this newer version, but it's still under development. So I think it'll be super useful, but at this point in time, I'm just kind of like putting it way to the side because I don't want to go down that rabbit hole yet. And then there's a value channel, which is a single value, such as genome size or coverage or something like that. And then in process, like I said, it's just where you execute all your tools and analysis. So let's say you have the simple workflow where you just want to assemble a genome, and then you want to remap those reads to the assembly just to see how well was this assembly or something. So naive me creates a workflow like this, where I have two FASTQ channels, one FASTQ channel. These are copies of each other. They're the same exact thing. But since I can only use a channel once in Nextflow, I have to create the channel multiple times of the same inputs. Afterwards, if y'all want to see how crazy this becomes, I can show you with the Bactopia. You end up putting like 10 different channels that are the same exact thing. But yeah, so FASTQ channel gets assembled, and then that assembly gets put into outputs, but also the FASTA file gets put into a FASTA channel, and then you take these two and you map the FASTQs here to the FASTAs here, and then you get your outputs. Great. But naive me again ran into this issue where the first in, first out can really cause problems for you. So let's say you have three samples, red, blue, and green. So they're denoted by your FASTQs are these red square, blue square, green square. So you can see these two channels are exactly the same. They're just duplicates of each other. But this FASTQ channel went to the symbol genome, and the symbol genome, the green one finished first, and then the red one, and then the blue one. And so now you're going to map these together, but now you're mapping the red FASTQs to the green the FASTA files, and the blue FASTQs to the red assembly and the green. And so now you have this mismatch. And I originally had this issue in Bactopia where I was like, why is this genome mapped to this one? Like, why did it select this reference genome for variant call? And it's like, they're so far apart. And then I was like, all right, let me create these little like debugging things where basically when a process runs, it'll say this sample is going to this sample. So basically I was outputting the name of this red square and the name of this FASTA, so I could find the mismatch. And it's like, crap. Well, that's the problem. So what ends up happening is you have to carry these basically these channel inputs across processes, so that way, that way they stick together throughout the pipeline, and which is kind of a hassle, but for the paranoid people like me, it's refreshing to know that X, Y, and Z are sticking together throughout the pipeline. So yeah, when you do it this way, you're basically saying also keep these FASTQ channels going, so that way they stick together here and then they get passed. And now this process knows, hey, process these two together, and now you get what you're going for. So yeah, Bactopia carries those throughout. Bactopia also uses process templates. So when you start making Nextflow workflows, they get pretty high in the line counts, which kind of becomes a hassle to sift through. And so Nextflow allows you to create, they call them templates, but they're basically external shell scripts that you can use to tell the process, hey, when you run, look at this shell script and run what's in here. It is kind of templated. You can use Nextflow variables inside the shell script and all that. But it really helps debugging a process, because now, for example, Bactopia includes 25 different processes. So that's 25 different versions of this right here. So it becomes a hassle if you have to sift through all these lines to find that one little section that has the problem. Whereas if I know it happened in my assembly process, I can just go to the assembly template and figure out where the bug is. And like I said, it reduces the main workflow line count. Bactopia alone is 1,800 lines, which is, to me, a lot of that is white space and just dealing with Nextflow's just structure. But that doesn't include the thousand of lines of code that's in the template file. So that's your actual shell commands. And 600 of those lines is just the usage in Nextflow. A few warnings I have about Nextflow is it does have some features that are missing, or at least I personally think are missing. One is a command line argument, like parser. Like that to me is just like, oh, I wish, when I gave these commands or these parameters, it was easy to just automatically check them. Verify that this parameter is an integer and verify that this one is a float. And so you end up having to do that yourself. It's kind of like, yeah, you don't trust the user type thing. You basically have to verify each parameter is what it's expected to be. Whereas if there was a command like argument parser, it would do that for you in the background. There is no dry run feature, and it would be nice sometimes to just, before you're about to launch this large job, that you could just say dry run and it'll say, this is what I'm going to run, instead of running it and then figuring out what's going to run. But that's likely not going to be possible the way Nextflow is created. The main developer also said it's way easier to just create a small test data set and just run it and see what happens, versus because of the way Nextflow is, it's event based, so that those channels have to be filled for the process to execute. And the error messages are sometimes very hard to decipher. It just blows up and you're just like, what just happened? And then you're trying to sift through it and you're just like, what just happened? Like there's no details, or there's just stuff missing, or just they're not always the best. And it's just, over time, you just kind of slowly, I think we all just kind of do this, to try to find the point where it breaks and just forcibly break it, just so you can find a starting point to find how to fix it. Also, the resume function is sometimes easy to break. So Nextflow does resume, which tends to work great. But if you run the same multiple workflows in the same directory, you might end up breaking the resume function for another workflow, because it tends to only resume the last executed workflow. So if, for example, if you ran Bactopia, then you ran a Bactopia tool, but you're like, oh crap, I messed up something in Bactopia, let me resume that real quick. You may have just broken it because the two Nextflow workflows kind of overwrite the cache of each other. And this is one that tends to step on the toes of others. By default, Nextflow uses all available resources. So yes, it has very good support for clusters and all that. But if you're on a shared resource, like some some nice little 24 core server or something, and you execute your Nextflow workflow, it's going to use all 24 cores unless you tell it not to. So yeah, it's one of those where it's just something to keep in mind. It doesn't do that if you're on a cluster, because if you're on a cluster, you have to say, I want this amount of resources. So what's next for Bactopia? I'm hoping it'll become a useful resource for the community. It'll really help as we get more users, just to polish it and get a sense of what people want. One thing that I didn't really highlight in the Bactopia data sets is there's this opportunity to create a curated set of species-specific data sets. So for example, someone who is an expert on the biology of like Haemophilus influenza, they may know the genes that other biologists are going to be, or other influenza people are going to be interested in, and they can create the set of genes that can be put on something like a GitHub repo or something that the Bactopia datasets can then import. And now you have this curated set of reference genomes and genes that most likely they're going to want to know details about. And I'm planning on creating more Bactopia tools, such as Mastery, HiCap, Backdate, parcer, and all those. So if you have ideas for tools, but the inputs may not be available in Bactopia yet, let me know, and I'm happy to see if we can implement that. For example, PopPuck2 takes a specific sketch, which is not created. It can use mass sketches, but it also has its own sketching library that I plan on adding into my Minmer sketching step, just because now I can directly take those inputs and use them downstream. I think eventually long read support will be added, but right now, honestly, it's just moving way too quickly to like nail down to the ground. Like yes, I could include it, but next month there's going to be a better assembler or a better mapper or whatever for long reads. So I still think that's probably a good few years before it's kind of like long reads, or it's in its groove where you have standard set of tools and all that. And then, yeah, we plan on processing the 70,000 plus Staph aureus genomes all on AWS, which will be the basis for our Staphopia version 2 web app. So those outputs will all be available. Acknowledgements, Tim Reed, who's on this call. I'd also like to, for the Bactopia preprint, Monica Farley, Oliver, Torsten, Narcisco, and Michelle. Oliver, Torsten, and Narcisco have tools very similar to Bactopia, which I have in the slides towards the end. After this, please feel free to check out theirs. Oliver's is ASAP, Torsten's Noliber, and Narcisco's is Tormes. And in the preprint, we kind of highlight the similarities and differences between our three tools, or our four tools. And then, yeah, Bactopia tool feedback, those people who have slogged through the early days of, this doesn't work. There still be those cases where this doesn't work, but I'm happy to fix those now. But Takira, Ashley, Taj, Ahmed, Curtis, John, Matt, Michelle, Sean, and Emily, and fund and sourced NIAID, Amazon, and CDC Emergent Infections Program. These are the tools, alternatives to Bactopia, but I'll leave this here, and I'm happy to take any questions or any offer, anything people have.
Yeah, thank you, Robert, for that talk. That's such a comprehensive talk from like the use case to the technical aspects. So I think there's going to be a lot of conversations going on. And also in the chat, a couple people have posted questions there as well. But yeah, and so yeah, thank you for giving that talk. I think the public health use cases are pretty apparent straight on. and I had a question about, so one thing that we're considering here in, Virginia Public Health Lab is especially instances where there's like interstate outbreaks. Right now, we rely on the nationally curated database for the enteric pathogens. There's things like the bionumeric side of things. For other things, we also have NCBI's pathogen detection software, where they automate that cluster detection happening there. And once our read data gets uploaded to NCBI, then it also gets analyzed in pathogen detection in that way. But the issue there is that there's like a 2 to 3 day turnaround time from when we have data and when we find out whether or not it's related to a species, high genetic relatedness to another species from another state, because we don't have that data. In this case, feasibly, I'm thinking we could use Bactopia to just get all the reads from NCBI for that pathogen of interest, maintain that mashed database, and then run that locally immediately. Is that?
Yeah.
Oh man.
Yeah. There's nothing that would stop you from giving your own mash sketches and all that to run these samples against. Yeah, that's the fun thing I think about Bactopia is this ability to give your own data sets. You're not stuck with what's coded into it.
Sure.
And that also kind of puts the responsibility of your own data sets on you and not me.
Yeah.
So it's kind of nice to have that door for you to be able to walk through and do what you need to do, versus saying, "Hey, Robert, will you open this door for me?" and that type of deal. So yeah, there's nothing stopping you from doing that.
Okay, I think that's like one of the first use cases in my head that popped up that I think a lot of us would benefit from. And then also in the chat, so if you see from Lee, you had a question about the rating system of I think there was Lactobacilli submissions. How do you categorize gold, silver, and bronze and rejections? Is it quantified automatically? And do you have specific thresholds per taxon? And if so, because if there's more to that, go ahead and answer that.
Yeah, so how do we categorize? It's a combination of read links. So we're dealing with the Illumina data, but we're also dealing with public data, which still includes some of those older Illumina sequences, which were only like 33 or 36 base pairs. So like we're including everything in this analysis, at least. One thing I should say though is these parameters are adjustable by the user. So if you don't agree with my thresholds, guess what? You can change them. Like, done. But I use per read, average quality, read links. This time I did number of contigs, just because it's nice to quickly see this assembly has 1,000 contigs, and there's something going on there when the rest just have less than 100. And what's the fourth one? Coverage. So and then by default I use 100x coverage, quality, a mean quality score of I think it's 33, and read length of greater than 90 base pairs. That's just to cover those random ones that are there, mostly 100, but after cleanup the medium or the mean may have dropped. Let's see. Yeah, is it quantified somehow automatically? It's based on those 4 criteria, which you as the user can adjust. Do I have specific thresholds per genus or taxon? Nope, because as the user, I think that should be up to you. And just because for Staph aureus data, I know like what kind of looks good and what doesn't, whereas tuberculosis, I have no idea. Whereas somebody may be like, yeah, you probably don't need this much, but so you could probably adjust those. If so, because part of the Bactopia mantra is try to go with public sources. Are those obtained through public channels somehow? So yeah, this is where it would be nice if through the data curation step is if you use a specific bug or you analyze a specific bug and you find that these parameters are the better thresholds through this Bactopia datasets curation. You can add that to the README where it's like, we find that these are good gold, silver, bronze cutoffs, and these are good cutoffs to exclude. So it's kind of a combination of, I've kind of set the bar, and as the user, you get to adjust the bar to fit your needs. The other question, have you tried analyzing CREs with Bactopia? Yes, I have, but that was I ran it and passed it along, and they didn't come back to me with problems, so I assume it had what they needed. And would you suggest using Bactopia or over Staphopia version 1 for Staph aureus? Yes. If you are currently using Staphopia and it's working for you, continue using it. But I will primarily be working on Bactopia, which I also include a tutorial in the documentation to replicate Staphopia using Bactopia. So that's a good stepping stone. And then I think the Staphopia, the next change that I will do for the current version 1 is just implement the table to ASN forever bit, just because the last few years that's been the only change I've made to it, is going in there and making sure table to ASN has been updated due to its expiration. So if it's working for you, continue to use it, and if it does what you need it to do, go for it. But if you get to where you need to start adjusting things, yes, switch over to Bactopia.
I've got a question for you. Awesome presentation. This is really, really an impressive project.
Thank you.
I'm thinking kind of in the perspective of a state lab that might not have access to Linux or a Mac, and maybe they have a Windows machine where they could install